Good evening, everyone. Welcome to you all to the week 4 live problem solving session on NPTEL Bioinformatics Algorithms and Applications course. So, as usual, in this question, I will be discussing summary about the week 4 lectures of the Bioinformatics Applications and Algorithms course. Then we will be trying some sample problems on phylogeny. So we have started with the concept of sequence alignment, and in that we started with a pairwise alignment, which is a type of alignment that involves aligning two sequences to identify the optimal pairing of the sequences. This pairwise alignment is based on a scoring system that assigns positive scores to matching characters and negative scores to mismatching characters and gaps. The main objective of pairwise sequence alignment is to obtain highest possible score, which indicates the degree of similarity between the two sequences. Second, we have a concept called multiple sequence alignment, which we had a brief intro. So multiple sequence alignment involves aligning multiple, that is 3 or more biological sequences to achieve optimal sequence matching. So they are used to identify the conserved regions and to construct phylogenetic trees, which is our main concept today. These phylogenetic trees helps us to understand the functional and evolutionary relationship between different species or groups of organisms. Okay, to recap the pairwise alignment, we have started with the dot matrix method, which is also known as a dot plot method. It is a graphical method of sequence alignment that involves comparing two sequences by plotting them in a two-dimensional matrix. In a dot plot, if a residue in one sequence matches a residue with other sequence, a dot is placed. So if we have similar residues matching, a dot is placed. Otherwise, the matrix position is left blank. So if we are comparing two highly similar sequences, the dot plot will display as a single line along the matrix main diagonal. However, when the sequences are less similar, the dot plot will be more scattered with few diagonal lines indicating the sequences are less similar. Then we also started with the dynamic programming. So in dynamic programming, we have came across Needleman and Wunsch algorithm, followed by that Smith-Waterman algorithm, that is global and local alignment algorithms and how to solve a local and global alignment. And then we also had introduction to fast and blast, which are word or k-tuple methods. They uses a heuristic algorithms for the database searching. So fast and blast are the fast methods for aligning two sequences. So they begin by identifying short identical sequences known as words or k-tuples, and then uses dynamic programming to align these sequences based on these words. So these are recap about our previous sequence alignment methods. Now we are starting with the multiple sequence alignment. So multiple sequence alignment can be performed using exhaustive or heuristic approaches. In exhaustive algorithms, we examine all the possible alignment at once. Whereas in heuristic, they are also known as a progressive methods, and which are tree-based algorithms, wherein we follow a stepwise assembly of multiple alignments based on a pairwise similarity. Commonly used algorithms for multiple sequence alignment include progressive alignment methods like ClustalW, MAFT, tcoffee, as well as iterative methods such as muscle, SciBlast. So here each algorithm employs different strategies to generate alignments and balancing computational efficiency with the alignment quality. With this, I have few questions on multiple sequence alignment just for a recap. So how many sequences are needed for a multiple sequence alignment? those who are joined late, I'm just summarizing for them. So I started with a recap to the previous sequence alignment methods, that is dot matrix and dynamic programming, and then I started with the multiple sequence alignment. So this multiple sequence alignment is an important step for understanding the phylogeny or developing a phylogenetic tree, which is our concept of discussion today. Thank you for your answers. Yes. For a pairwise sequence alignment, we need two sequences to align. When it comes to multiple sequence alignment, we need three or more sequences to generate a multiple sequence alignment. So this multiple sequence alignments are used to identify the conserved regions and also helps us to understand the functional and evolutionary relationship between different species or a groups of organisms. Next, I have one more. In an here MSA stands for multiple sequence alignment. So in a multiple sequence alignment, what does a highly conserved region indicate? So across the multiple sequence alignment, what does a highly conserved region or a highly similar same region indicate? Yeah, in a multiple sequence alignment, a highly conserved region is indicated by indicates a low sequence variability. That is, they are similar, same across. Okay. So moving on to the concept of phylogenetic trees. Phylogenetic trees are diagrams used to represent the evolutionary relationship among the group of organisms or sequences. So these trees illustrate the evolutionary history by depicting the branch pattern that results from the divergence from a common ancestor. Just like comparing this phylogeny to a family tree. So it gives divergence of the offsprings from the parent. So these phylogenetic trees are fundamental tools in evolutionary biology, genetics, ecology, and other biological disciplines. So just like phylogenetic trees here in machine learning in other concepts to depict the relationship between the starting point and end point, we have concept of decision trees, random forests, which are tree-based methods. This is one of the similar tree-based method to understand the evolutionary relations. So let's try to understand a phylogeny tree. So what are the terminologies used? First, let's start with the nodes. Nodes represent the hypothetical ancestors or a common ancestors of the organism or a sequences being studied. Each node represents a point in evolutionary history where lineages diverge into separate branches. So the branch splitting point is known as nodes. So nodes are termed as points where branches split, as shown in the image. Next we have branches. Branches represent the lineages or the evolutionary paths connecting the nodes. So the length of the branch is often proportional to the amount of evolutionary change that has occurred since the divergence from common ancestors. So we have trees with branch lengths and we have trees, unweighted trees and weighted trees. We'll come to that concept later. Next we have a root. The root of a phylogenetic tree represents the common ancestor of all the organisms or sequences, including the analysis. It is usually placed at the bottom of the tree and represents the oldest point in evolutionary history. Then we have tips, or the other name for these tips is terminal nodes. These tips or terminal nodes represent the extent or observed organisms or sequences in the analysis. Each tip corresponds to a specific species or individual organism or a sequence being studied. So initially we have come across branch length. So this branch length represent the amount of evolutionary change. So here evolutionary changes means uh, number of genetic substitutions or sequence differences that has occurred. So these lengths are often measured in units such as substitutions per site or time units. Similarly, we have some other terminologies such as outgroup. So a outgroup is a part of a tree or a taxon, that is a single organism or a group of organisms that is known to be distantly related to a taxa being studied. So these trees can be constructed using various methods such as distance based methods, maximum parsimony methods, and maximum likelihood methods. Okay, so in some cases we see an outgroup may be included in an phylogenetic tree. So a outgroup, now when we construct a tree here, for example, uh, CDE are kind of similar in the evolutionary concept, and AB are a little distance when we compare with the CDE. So an outgroup is a taxon, that is single organism or a group of organisms that is known to be distantly related. So I told branch length will represents the evolutionary change. So which are distantly related to other taxon. Like for example, if CD is E is anything of tax of my interest or organism of my interest, then I can tell AB could be the distantly related organisms to them. So this helps in which direction the inferring the direction of evolutionary change. Like we can tell the outlier or the outgroup. Like outliers are outgroup here. Yeah. Phylogenetic trees can be constructed by various methods such as distance-based methods, maximum parsimony, and maximum likelihood. So these methods rely on different models of evolution or different types of MSAs and assumptions about the evolutionary process to infer the most likely tree topology. So next, we will be entering into a concept called rooted trees and unrooted trees. So rooted and unrooted trees are two fundamental representations used in phylogenetic to understand the evolutionary relationships among the organisms. In these two trees, the difference lies in whether the common ancestor of all the taxa is explicitly shown or not. If it is shown, we call it a rooted tree. Like if you show the root node, we call it a rooted tree. If you are not showing, it's a unrooted tree. So in rooted tree, a single node called the root node, here Z, which is typically placed at the base of the tree, indicating the direction of evolutionary time with branches diverging from it. So from Z we have A, from A again we have branching to B and E. So that. So these rooted trees provide information about the both relationship among the taxa and the directionality of evolutionary change. Next, we have an unrooted trees. In these unrooted trees, the common ancestor of all these organisms is not explicitly shown. Instead, this tree only shows the relationships among the taxa themselves. That is, B and D are closely related, and A and C are closely related. So these unrooted trees consist of a branching patterns representing the relationship among taxa without indicating the direction of evolutionary change. But they are still, since these unrooted trees do not provide any information about the direction of evolutionary time, still they are valuable for visualizing the evolutionary relatedness among the taxa. So then a question comes, how to select a rooted or an unrooted tree? So this depends on specific goals of the analysis and the available information about the evolutionary history of the taxa under study. So rooted trees are preferred when directionality of evolution is known or when the inclusion of an outgroup allows for a rooting the tree. So unrooted trees are used when the focus is solely on understanding the relationships among the taxa without making assumptions about the direction of evolutionary change. So here, both the representations are important tools in phylogenetics and provide valuable insights to the evolutionary process shaping the biodiversity. Okay, before I'm moving on to some mathematical formulas, uh, let's move into one mathematical concept about the factorial concept. So if I tell n factorial, like, so it is denoted by n followed by exclamation mark. So which is the product of all the positive integers less than or equal to n. For example, here n factorial, which is n followed by exclamation mark, is equals to the product of all the positive integers which are less than or equal to n. So given this introduction to the formula, so can you guys calculate what is 2 factorial, 3 factorial, 4 factorial and 5 factorial? I've seen some answers coming up. Yeah. So when we calculate a factorial, so we start from the highest number, then keep on multiplying with the integers which are less than the initial number till we reaches 1. Okay, moving forward, yeah, here is the answer. The 2 factorial will be 2 into 1, which is 2. 3 factorial will be 3 into 2 into 1, so value is 6. 4 factorial will be 4 into 3 into 2 into 1. So I'm just following the formula here to develop a factorial. And 5 factorial will be 5 into 4 into 3 into 2 into 1, which is 120. I hope everyone are clear with this concept. So based on this concept, this understanding, we are trying to do something in phylogenetics now. For example, given 5 organisms or 5 taxa, can we tell how many types of rooted and unrooted trees can be constructed? So that is our next concept to study. So given n organisms, how many trees, rooted and unrooted, are possible? So here, so how many rooted trees are possible with 3 nodes or 3 taxas, you can consider them. So you can use this formula. So where n stands for number of nodes, three nodes, so we can construct three rooted trees and a one unrooted tree. Sorry for the confusion the last. So moving on to next methods, which are construction of phylogenetic trees. So first and mostly widely used method is distance based methods, which involves calculation of a distance matrix. So under the distance matrix, first method today we're going to deal is with the neighbor joining method. So which constructs a tree by iteratively joining the pairs of sequences that are mostly similar until a complete tree. So it starts with the combining or joining two sequences that are most similar until we add all the organisms into the tree. So initially it starts with a star-like tree where all the tags are connected to a single central node, and at each step it identifies the pair of taxa with the smallest pairwise distance and then merges them into a new node connected to the central node. So this process continues iteratively and gradually building the tree until all tags are connected. Okay, so first we'll see this method how. So before moving on to this neighbor joining method, given a sequence alignment here, how a distance based matrix is constructed. So here the distance is nothing but the number of mismatches. Let's consider the distances to be the number of mismatches. So in order to extract the number of mismatches, first let us extract the possible pairwise sequence alignment for the given multiple sequence alignment. So I have given a multiple sequence alignment of ABCD organisms. From this multiple sequence alignment, I have extracted all the possible pairwise alignments. Given AB alignment is similar to BA alignment, AC alignment is similar to CA alignment. So these are the possible pairwise alignments that could be extracted. Then we try to find the mismatches in individual pairwise alignment. So here I have highlighted them in bold. So in between A and B, we have only one mismatch, that is at the last place, CG. In AC, we have three mismatches. So now knowing the number of mismatches, we'll try to build a distance matrix. So along the diagonal, I have added zeros because when we try to take similarity between A and B, mismatches between A and B, there are A and A, there are no mismatches. B and B, no mismatches. C and C, no mismatches. D and D, no mismatches. So I have added along the diagonal all zeros. Then number of mismatches between A and B, 1. Similarly, I have built the complete distance matrix. Further, there are several formulas that are available to convert them into this evolutionary rates, evolutionary time. So based on our needs, several formulas are available. But today, I'm just considering their mismatches as such. And when we observe this matrix, the matrix above the diagonal is similar to the matrix below the diagonal. Diagonal is 0, 0, 0, this line. So these are the symmetric matrices. This is a symmetric matrix. Then see, I told this neighbor joining method starts with a star-like tree where all the tags are connected to a single central node. Now at each further steps, we need to identify the pair of taxa which are similar, and then we need to merge them into a node, single node. So here I took five sequences A, B, C, D, E, and the distance matrix of them is provided. Then based on this distance matrix, what are the two what is the smallest distance, and that is between what? So here we have two options. AB can be considered or DE can be considered. So we can start with the any step. So I'm starting merging A and B into a node AB here. Since the distance between them is smallest, so we start with the smallest distance. Similarly, one can start with the DE also. So once we have formed a new node AB, which is diverging point of A and B, we try to reconstruct the scoring matrix using the formulas. So where we take sum of the distances of A to other nodes and B to other nodes and subtract it by the distance between A and B divided by 2. So this gives the new distances. So this matrix is constructed according to the formula shown here. For example, when we take the distance between the new node A, B and C, so first we sum up the distances of node A and C with the node C and B, so which is 3 plus 3. Then we subtract the distance between A and B, that is 2. Then we end up having a value of 2 here. So the distance between A and AB, node new AB and C is 2. Similarly, we calculate for other nodes, and the nodes such as CD, CE and DE, there is no change, the values from the previous step. So when we observe here, again we have 2 as a smallest value, which is between A, B, node combined node AB and C, and D and E. So here again I have taken ABC. So a new node has been formed, which is a combination of A, B and C. And similarly, the distances of the new node ABC with D and new node ABC with E has been calculated. And finally, we join D and E to complete the tree construction. This is how a neighbor joining method takes a progressive way of identifying each pair of taxa with the smallest pairwise distances and then merges them into a new node. Finally, when we see D and E are still together, initially also we see them to be the closed. Even when you start constructing from D and E, the process will be D and E will be close to C, then this combined one will be close to A and B. This is how, when we consider D and E, the method progresses. Next, to start with the uh, UPGMA method, we have one more method, which is UPGMA method, where UPGMA stands for unweighted pair group method with arithmetic mean. This is also again a distance based method. So it starts by clustering the taxa into the groups based on the pairwise distances, then repeatedly merges the closest cluster until a complete tree is formed. So here I have taken one common example for neighbor joining and UPGA. So I constructed a distance matrix again. So when we observe the distance matrix, the first step is to find I and j with the smallest distance. So when we scan through the matrix, we have set of numericals where 1 is the smallest value. So 1 is the difference between A and B and E and D. So since we can start from anyone, again I'm starting with A and B. So I have grouped A and B together. I join A and B together. Then we need to reconstruct the distance matrix. So here we take the average distances between, here the formula has been changed. So here what we do is, when we want to extract the distance between the combined node A, B and C, we take the average of the distances between AC and BC to reconstruct the new distance matrix. So in the new distance matrix, we see the nodes D and D are having the smallest value. So we join them. Then again we try to reconstruct the matrix using the same average formula. Then in the new matrix, we have smallest value between the combined node A, B and C. So we have joined C here. Here the values 0.5, 1, 1.5 denotes the branch length. Similarly, we keep on recalculating the distance matrix and then keep on constructing the tree. Finally, we lead to a final tree. Apart from these methods, we have other two methods which are maximum likelihood method. So maximum likelihood is a probability based method that estimates the parameters of a sequence evolution to find the tree that maximizes the likelihood of observing the given data. So this is based on probability of substitutions. Then we have maximum parsimony. So this method seeks to find a tree that requires the fewest evolutionary changes or the simplest explanation to the observed data. So this is what explanations I have for today. So any doubts in this?
Sir, these so many methods you have described very nicely. Is there a recipe when to use what?
In general, based on all the softwares are inbuilt with all these methods. So distance-based methods are highly used now. In general, neighbor joining is the most preferred because it is an like these are a progressive methods which starts from bottom-up approach. Other two methods are top-down approaches. So bottom-up approaches are preferred. Okay, since that I have explained what are different terminologies that are involved in a phylogeny, this is a match the following kind of question. Okay. So what do a root represent in a phylogenetic tree? So this is kind of a match the following. You can consider left side having numericals like 1 2 3 4, and the right side having ABCD options. So you can tell whether the root 1 is related to A, B, C, D accordingly. Yeah, okay. Thank you for some answers. Yeah, as we discussed, the root of a tree represents the common ancestor of all the organisms or sequences included in the analysis. So, common ancestor stands for origin of evolution. Thank you for your answers. Next, we have leaves. The other name for leaves I didn't mention is about tips or terminal nodes. So that stands for current organisms. Branches show the relation between the organisms, and branch length gives the evolutionary time. So here the branches show the relationship between the organism, whether they are closely connected or far. Then the branch length gives the evolutionary time. Given the professor has explained about the Newick notation, so try to provide the Newick notation for a given tree. So for the tree B, I have given a hint below. So if the tree is scaled, I told there is weighted and unweighted. So this is the difference between the weighted and unweighted trees. So the branch lengths are indicated immediately after the taxon name. So when you are generating a Newick format for the given trees, okay, to solve these questions, let's move on to the Google Jamboard. So let's try to solve for the tree A. So we try to group from the exterior ends, that is from the end of ABC. So first we try to check what are the closest ones. So BC is close. Then the next closest one to BC is A. And next is DE. So in other case, it could be written like A, B, C. So which represents this part. Then this is closely related to a group of tags or DE, which represents this part. So this is the unique format. So this is the format here. Now let's go and solve for the tree, scaled tree. So in a scale tree, whenever we are writing a taxa or an organism name, then we represent the length at the same time. So A2. Then we have B1, C2, which is closely related with D 1.2, sorry, E 2.5. In some cases, just to differentiate between the taxon name and the distance, a semicolon is being used like this. Just to separate the taxon names with their branch lengths, not to confuse for each others. A semicolon based method has been introduced.
Sir, in this diagram, why BC is taken? Why not A first and then BC, then D and E? Why BC suddenly appeared? Instead of that, actually ED should appear, then CB should appear, then A should appear. I don't know what is going on.
Generally, I told it starts from the exterior side. So it starts from ABC side. If you see the image, ABC is on the top. So as you move down along the image, we try to group the first node we see. What is the first node we see here? It's a node combining B and C.
Why not A? A and then BC?
No, that is also right. No, I explained here. No,
Okay.
I explained the previous case, not in this case. I think so.
Okay.
No.
Okay.
Yeah.
So in the final exam,
Yeah.
if we write whether A and after BC and DE, or in the other fashion, the marks will be deducted or it will be same?
Yeah. So I think you are taking up only the MCQ based ones. So, and also when you write this, we have this text mapping software available with NPTEL. So in whatever case you give, all the answers will be considered as correct.
Okay, thank you so much.
Thank you for letting me know that mistake. I'll correct it and upload later. So given UPGMA method, which of the following stands untrue about the UPGMA method? So the first option says the simplest clustering method is UPGMA, which builds a tree by sequential clustering. The option B tells given a distance matrix, it starts by grouping two taxa with the largest pairwise distances in the distance matrix. Option C tells the distance between this new composite or the combined taxon and the remaining taxon are calculated to create a reduced matrix. Then the option D tells the grouping process is repeated and another newly reduced matrix is created. It's B. So we start with the smallest pairwise distance, not the largest pairwise distance. So these are the steps. So it starts with the calculate the pairwise distances. So pairwise distance is nothing but the number of mismatches when we compare a possible pairwise alignments in a given multiple sequence alignment. Then here the option 3 tells we cluster the two sequences with the smallest distance to each other. Further have some more. A bifurcating branch point in a phylogenetic tree is known as. The screen is visible. The, please check your connection. Yeah, the diverging point is termed as a node. So given a peptide and weights of individual amino acids, what will be the molecular weight of the peptide? I think introduction like this could be helpful. given the peptide sequences and the molecular weight of individual amino acids, what will be the molecular weight of a peptide? Okay, let me. So this is the given peptide sequence. So when we sum up the individual weights, finally we end up with 870. And when we observe them, a peptide bond can be formed in between L and E, E and A and D, D and S and D, and E and Q. So whenever a peptide bond is formed, H2O is released. That is dehydration reaction. So the molecular weight of H2O is 18. So overall, the sum of the molecular weights of amino acids is turned out to be 870 minus molecular weight of number of water molecules lost. So N into 18. So here we are losing 6 water molecules. 6. So the final answer will be 762. So when a peptide bond is formed, a water molecule is lost. So for each peptide bond, we subtract the weight of a water. So given a pattern, try to identify what are the following options will match with the pattern. Okay, I'll give small hint. and how these patterns are read. So given this is the pattern, the square brackets denote for any of the mentioned ones. So in the first place, we'll have any one of P, L or A. So in the first place, it could be P, L or A. X denotes for any amino acid. So let's keep it X only. And next, since it is directly given G here, we are having G. Then the flower brackets denotes except K and P, anything can become. So here for next two places, K and P are not allowed. Finally, we have Y. So based on this, we'll try to answer this question. So in the first one, since this is not a valid one because X cannot come, some amino acid should be replaced here. We don't have any single letter code for X, and 2 is a numerical. The first option is wrong. Then coming to the second option, the first condition is satisfied. In the first place, we have P, and in second place, we can have anything. So again, we have P followed by G, but in the fourth and fifth place, we should have amino acids which are except K and P, but we have K. So the option B is also wrong. Option C is wrong because in third place we should have G. Then left over with option A. First place we should have either P, L or A. We have L followed by X. So we have A. Then we have G. So instead of K and P, anything could be added here. So RA we have followed by Y. So the answer is D. Yeah, so these are some tasks. So everyone clear with this pattern question, right? Any doubts or any doubts in the whole? So today I'll be demonstrating about multiple sequence alignment tools, how to extract multiple sequence alignments and visualize tree. Yeah, before moving on to that, everybody are able to access my material, no? The Python codes or the sample data files that I have sent. Please reply yes or no in the chat box. Thank you for your answers. Moving on to next thing. So we have discussed some of the multiple sequence alignments. So when we observe the current literature, we have some, one second, progressive alignment methods widely used. So the widely used progressive alignment method is cluster omega, or we have previous version cluster W also. So cluster omega. So cluster omega is available in EMBL EBA site. So we'll look into this. So here in the description it is given that cluster omega is a new multiple sequence alignment program that uses seeded guided trees and hidden Markov model profile techniques to generate the alignments between three or more sequences. So when we see, the first input option is the sequence input. So here, so under the input option, we can either give in GCG format, FASTA format, GenBank format, which is a flat file format. And then we can select what is type of a sequence, protein, DNA or RNA. So for today's, I'm using the use example option. So this is a predefined set of sequences that are given by developers. So here I have a sequence of human hemoglobin, mouse hemoglobin, then I have some coffee hemoglobin. Then once it is done, then we select with the output format, like output format, nothing but cluster W with character counts, or cluster W without character counts, FASTA format, and we have some other Nexus, Phylip, and other formats. Phylip is the standalone version of multiple sequence alignment tool as well as phylogenetic tree construction tool. Then I'm giving demo like cluster omega demo and then submit. In the more option, we can see, we can also change distance matrix, whether we need a guide tree or not. And we can also select the number of iterations for hidden macro model. So I'm not changing anything. I'm just clicking on submit. It will take 1 to 2 minutes for the job. Yeah. So when we observe the results page, we have several tabs available in the results page. So the first tab is alignment tab, next tool output, guide tree, phylogenetic tree, results and result file formats. So under the alignments tab, we can see the color coded multiple sequence alignment that is provided. So most of these colors are selected based on the physiochemical properties of the amino acids. For example, similar color for arginine and lysine. Similarly, when we go and check the tool output, a proper multiple sequence alignment with the character count is given. So here the star denotes exact match. So this is match in all the three places, and the semicolon denotes similarity. Then we have a phylogenetic tree. This is the phylogenetic tree. This is a just reference phylogenetic tree that we get from these multiple sequence alignment programs. Followed by this, we have one more software, MAFT. You can type MAFT alignment. So under MAFT cbrc.jp, we have MAFT alignment software. So MAFT has a downloadable version, standalone versions, and online version also. They have a separate release for Mac OS. They have a separate for Windows, Linux. So I'm using the online version for today. Similarly, I'm inputting the example sequence. So these are set of FASTA files given, protein FASTA files. So I'm copying them, Control A, Control C, then pasting it here. So additionally, when we are uploading a large number of sequences, we can provide our email address. Once the job is done, the results will be mailed. I'm just submitting. So in the mapped results, we can see view the cluster format of alignment. Then also we can see the tree. So this is the multiple sequence alignment that we have obtained for the given input sequences. So all these bioinformatics tools come with an inbuilt example data sets which can be used for the understanding purpose. Then this is a distance based tree that map gives us. So either this NJ stands for neighbor joining and UPGMA method. So this gives this sequence uh, statistics. So in this we have so this is the phylogenetic tree. For the 17 input sequences. Here the bifurcating nodes are given in black dots, bright black dots. Yeah. Previously, someone asked for the outgroup. So when we see this top two sequences, okay, let me zoom a little. Top two sequences, these are completely deviating from the rest of the other sequences. So these two, SESA and Serenti RNA ligase, comes under the category of outgroups with respective to these proteins. Yeah, that's all for today. So I'll take some questions. If there are no questions, I'll try to demonstrate how to use a installed version of phylogenetic tree construction methods. So we have a software called a Mega software, which can be used phylogenetic. So they have available version for Windows. So here, based on your, once you go into this Mega software page, you can access based on your OS, whether it is Windows, Mac OS, Ubuntu, and other Linux versions, and then you can download and install. So once you install, the interface looks like as shown here. So we have align tab, data tab, models, distance, diversity, phylogeny, user tree, and other things. So first we select the data tab, then we try to open a file or a session. So already I have made a sample MSA file. This is a FASTA format file. We can visualize it here. Then once you input a file, whether it will ask to analyze or align. So I'm clicking on align. So these are the sequences that are available in the FASTA format that I have given. So this is the similar input file what we have used in the MAFT. Then I'm selecting the alignment tab. So here I have align by cluster W or align by muscle. So two options have given. So I'm selecting cluster W. So these are the some of the alignment parameters like gap opening penalty, gap extension penalty. Then click on OK. First the pairwise alignment for the each, all the combination sequences performed, then we get into a multiple sequence alignment. So this is the multiple sequence alignment. So one can, before generating the phylogenetic tree, we need to save this multiple sequence alignment. So to save this, I'll go to data, export alignment in Mega format, because again we are going to generate phylogenetic tree from the Mega software only. So I'm saving it in the Mega format. Then once I save this, I'll go into the phylogeny option. So under the phylogenetic methods, we have discussed the maximum likelihood method, neighbor joining method, UPGMA method, and maximum parsimony method also. So here I'm selecting neighbor joining method. Then this is the MSA Mega format file that you have generated. I'm selecting it, open. Now some options you see, then click on OK. The tree construction has been started and completed also. So this is the phylogenetic tree that we can see. So in different ways, the tree visualizations can be modified. So next, we can view as a rectangular, straight. We can also view it as a curved trees. We can also view these in a circular format. Yes, please.
I just wanted to know,
Yes.
after doing this type of analysis, will it write a sort of a paper or a draft that this species is close to this, etc., all that paragraph, instead of looking at all this data, will it generate automatically?
uh, Mega software doesn't do that, but we have other deep learning based tools coming up now, which does this based on the orthogonal representation of the sequence.
Thanks. Thanks.
So we have, apart from this, we have several tools that are coming up to have this visualization, to make visualization of this phylogenetic tree. Because now that we have taken only 17 sequences, still the tree looks kind of complicated. Wherein in some analysis to understand the evolutionary history or in genetic space studies, you need to align thousands of sequences. So that case, this makes more complexity. Where previously machine learning methods are there, currently deep learning based, uh, LSTM based methods and CNN based methods are coming up to give the visual summary of the summary from the visual interpretation of these phylogenetic trees. So that's all for today. Any questions I can take?
I think Harshit Singh has a question on the chat box.
Okay, let me open. Give me a second. Yeah, so this is an unrooted tree, and uh, the tree, when we are selecting the tree also, there's an option for us to select. So the bootstrapping data is a kind of clustering based method. So that helps us in giving the unrooted trees. Even we have branch test based method. Finally, I have some task questions. Yeah, these are some task based questions given for you to take home. things. So first one is composition calculation. Uh, One can use seek to feature tool that is available from our lab to calculate the composition, as well as for the molecular weight also. Seek to feature tool can be used. Then given the composition of group A, set of amino acids belonging to group A and set of amino acids belonging to group B, then calculating the standard deviation, what could be the amino acid sequence given in the question 1 belong to, then average hydrophobicity based question. So these task questions are made such a way that practicing them or solving them by your own helps you make you prepared for your end exam.
Can you please explain what is this group A first value and group B second value?
Yeah. So I have two sets of groups. So group A, group B. So the average alanine value from group A is the first value, and like for example 8.47, and the average value of alanine from group B is 8.95. And when you calculate the composition from the question 1, so you have three sets of composition values now. For alanine, three values from group A, from group B, and what you have calculated from Q1. So based on this, you can calculate the deviation of the calculated value from group A and the deviation of the calculated value from group B. So whichever the deviation is less, the sequence would, like this is for example for alanine. Similarly, you should repeat for all, and when you sum up the deviation, if the deviation value calculated composition is having less deviation with the group A, then it will be belong to group A. If it is having less deviation with the group B, then it belongs to group B.
So when we say deviation, we have to take a square of that, add all of them and take a square root of that or something like that.
Deviation stands for differences.